Welcome students. We have started our discussion with stem and we have discussed our root also. In that we have covered dicot root and then moving on to monocot root in this lecture. Now monocot root also, this monocot roots are basically, you can say they are fibrous or adventitious in nature. Okay, that means these roots, lot of fibrous roots will arise from the base of the stem and there won't be any a single main tap root system. Many of the roots will have a similar length and they will originate from a common point. Generally they are found at the base of the stem, but sometimes in other parts also, like for example in leaves also these type of adventitious roots can arise. Now when we cut the section of this root, either these roots or some other lateral branches which are coming out, these fibrous root all will have the same anatomical structure. When we will cut the anatomical structure, we will see it will also have a boundary. Okay, so that outer boundary is definitely, it is the epidermis, but in roots we call it with a different name. So it is, you can say rhizodermis or epiblema, or even you call it as piliferous layers. Now this will also have root hairs. Okay, so they will also be surrounded by root hairs. So root hairs are outgrowth of a single cell. They are not attached from the single cell, but they are itself the cell which have grown out in the hair-like structure. Now this epidermis will be followed by cortex. Now cortex will be more elaborate in case of roots and it will be more homogeneous in nature. Like in stem we have discussed like different types of tissues were there which were forming the composition of cortex. However, in this case you will have mostly they will be parenchymatous in nature. Okay, and you won't find any distinction of hypodermis and different cell layers like sclerenchyma or collenchyma. However, some small variations could exist, but as such in general you won't find these layers. Then in the cortex, after cortex you will have endodermis. Okay, and then endodermis will be followed by pericycle. Generally a single layered endodermis will be there. And then you have pericycle. Okay, this will also be generally single layer. Okay, and inside this also you will see vascular bundles. Vascular bundles in patches you will see. Okay, so this type of radial arrangement is known as radial. So as many rings, as many patches of xylem as phloem, means patches of phloem and xylem will be same. And in the center you will have pith. Okay, so pith will be quite elaborate and pith will be distinct as in case of root. So it will also be made up of parenchyma. Now in this vascular bundles are generally you can say it is 6 or more than 6. So better to draw more numbers of vascular bundle here. So vascular bundles will be 6 patches or more. So this type of condition is known as polyarch condition. They will alternate, see xylem and phloem, they will alternate in a ring-like arrangement. Now here you will see endodermis and pericycle. However, the difference between the endodermis means difference between the pericycle of monocot as well as dicot is that pericycle will not participate in secondary growth formation as it was in the dicot root, because dicot root are also quite wider, they are also thick and big, so they undergo secondary growth formation, and this pericycle layer is the one which after a certain period of time they start behaving like meristematic tissue, but in monocot root, so no secondary growth formation will be there. So this pericycle will not participate in such type of division. Now when we draw the cellular diagram, so here also you can draw it similarly, you can draw an outer epidermis, okay. Sometimes in case of these roots, some layers slightly become thicker and they are known as exodermis, but in most of the cases it's not there, it's not found. In some plant tissues it could be there. So that also we will discuss in some special case when we will discuss some different types of roots, different types of adaptations of root. So there we will try to discuss exodermis. So in this also this is an extension, root hair will be there which is an extension of. Then you will have parenchymatous, cortex, okay, and then you will have endodermis, which will be made up of barrel shaped cells and containing Casparian strip. Okay, so thick walled cells will be there on radial walls as well as transverse wall. These thickenings will be there. Okay, then this will be followed by pericycle, followed by xylem and phloem patches. See, in this pith is bigger, so this is also one difference between monocot and dicot root. So in this case also, see, you will metaxylem will be definitely towards the center and protoxylem on the outer side. and this type of arrangement is known as exarch, okay, because protoxylem is all on the outer side towards the epidermis. Pith will be there in the center, okay, and phloem patches will be there, and in between conjunctive tissues will be there. Okay, this way monocot root will be arranged. It is, you can see that difference is very less as compared to dicot root. However, slight differences in the terms of a bigger pith. The most important thing is that it will have a bigger pith, and the next important thing is that the polyarch condition. Okay, more patches of xylem and phloem will be there, which will be arranged in the form of a ring. Okay, and some small differences definitely can be there, but that depend on plant type. Now, this is also a diagram which shows you how there is a difference between dicot root and a monocot root. See here, you can see that the pith is highly reduced. Okay, pith is highly reduced, and many a times it is almost absent also. But in this case, you will have a well elaborate pith. Okay, however, the epidermis will be there. Sometimes, as I have told you, some layers of exodermis could also be there, but it is not very common. Then cortex will also be there, but cortex is quite elaborate in both the cases, and that will be parenchymatous in nature. Here also endodermis is there. You can see endodermis in both the cases. Then pericycle followed by pericycle. Okay, here also you will have pericycle. Okay. Then these are, you can see different patches of xylem and phloem. See, these are all vessels. Okay, so it's, when we draw a diagram, we generally show rounded structures which are basically the vessels of the xylem, because they have the most wider cavity, and when it is seen under cross section, you can see those cavities in the form of hollow tubes. So these structures are vessel. Okay, you can see smaller vessels are on the outer side and bigger vessels are on the inner side. However, the number of these patches are much more in the monocot as compared to the dicot. Similarly, phloem will be there in between these xylem patches. Okay, see, these are the phloem patches in between the xylem layer. See, this is the xylem and this is followed by phloem patch. Similarly, here also phloem patch is there and xylem is there. So, this is the diagram, this is the transverse section. Okay. Now, when we actually draw a cellular diagram, although it is a cellular diagram only, but there is also a way of drawing it, like I have shown you also in several cases, like when we draw an outline, okay, we generally try to make it a complete outline of the showing all the layers. Okay, generally like stem or root, you have a circular outline, but it's not necessary, but generally we try to make it an entire structure showing with line diagram the outline of all these structures. Okay, like that. Suppose if I draw a dicot structure like this, dicot stem, suppose if I am drawing, or suppose root also you are drawing. Okay, root also you are making patches. Okay. So when we draw the line cellular diagram, we take a portion of this entire circular structure and then we make a line diagram. Okay. Now in case of root, because since if you just take a, cannot show the entire arrangement of vascular bundles, so in that case we take a section of cortex covering the epidermis and cortex and then covering the entire stele. So here you can see that the entire stele is shown in a complete diagram, whereas when you draw in case of stem, like in dicot or monocot stem, these have separate individual vascular bundles. So we can clearly show the cellular structure of an individual vascular bundle. So we take a section which can have one or two vascular bundles in it, and then we can elaborate in and draw a complete cellular diagram. So here also this is a cellular diagram which is just showing the difference between monocot and dicot root. Now, development of lateral root. See, every root will have lateral root, whether it is the dicot root or whether it is the fibrous root or monocot root. See, lateral branches are definitely coming out. Here also so many lateral branches are coming out. So, these lateral branches will originate from pericycle, that we already know that pericycle will give rise to lateral branches. See the root apical meristem. This is only elongating the length of the root. However, it is not giving rise to lateral branches as was the case with the shoot apical meristem, where it was giving rise to leaf primordia. See, that all we have discussed, that this is the leaf primordia. So this apical meristem, when it was growing, it was outer tissue were giving out branches or leaf primordia. So that development was known as exogenous. Okay. However, in case of root, the pericycle, which is present in the vascular region, in the central stele region, okay, it is the outer covering of the stele region. From there, actually, lateral branches will come out. So, this type of origin is known as endogenous, because some tissue which is present inside the plant part will give rise to root, lateral root. Okay. And this type of development is applicable for monocot and dicot root both also. Like in monocot also, whatever lateral branches are coming out, they are coming out from pericycle. Now, what actually happens, suppose you have endodermis here, okay, endodermis layer is here. And then you have pericycle. Suppose pericycle cells are there, okay. So, what will happen in this pericycle layers? These layers will divide tangentially, okay. They will start behaving meristematic. They will start behaving like meristematic cells, and they will start division, and these will push the endodermis on the outer side. More further, more divisions will occur. More divisions will occur in this pericycle, forming more layers of cell, and they will push the endodermis on the outer side, and finally they will actually rupture the endodermis and on the outer epidermis or rhizodermis also, and they will come out from that. And as they are growing, they will also develop their own meristematic tissues, means they will be differentiated into meristematic zones. Okay, so you can see here that as these pericycle cells are dividing, they are pushing the endodermis, and they have ruptured the endodermis, and finally when they will move towards the cortex, they will also rupture them, and finally this outer covering also they will rupture and they will come out. and they will be also differentiated into the meristematic zones, that is dermatogen, perivellum and pleurome, that we have discussed in histogen theory also. You can name them as also protoderm. That all we have talked about many times. Then protoderm, then ground meristem, and then the procambium. Procambium was there, but in this case, this will give rise to the stellar system. Okay, this will give rise to the ground tissue, and protoxylem will give this protoderm will give rise to the epidermis. So in this way, the lateral meristem will be formed, and this will give rise to lateral root. So this was how a root in dicot as well as in monocot plant was developed, and we have also discussed how lateral roots were originated. Now let's try to discuss one very important concept, that is root stem transition of the vascular tissues. See, in stem, the arrangement of vascular bundles as well as the arrangement of xylem, like that is protoxylem and metaxylem, is entirely different if we compare it with the root. Now in stem you have conjoint vascular bundle and endarch condition of the xylem, okay, whereas in root you have radial vascular bundle and exarch condition. That means this type of arrangement was there, where phloem was there on the outer side and xylem was there on the inner side, and both were lying on the same radii. And in this case, when we talk about xylem, then protoxylem was towards the center and metaxylem towards the periphery. In root, you have a different arrangement, you have different patches of xylem and phloem, okay, and in that also you have exarch arrangement. That means protoxylem is on the outer side and metaxylem is on the inner side. Now see, this definitely when the vascular system is continuous, vascular system is of the root system and the stem system, it's continuous, it's not broken. So there has to be a transition zone or a transition point where there is a change in the vascular system, and you should also think that why there is a change in the vascular system. Why not root vascular system is continued in the stem also? Why it is changing? So the answer here is that like these conjoint vascular bundle actually provides a more organized and better transportation system, and with the evolution in plant tissue structural organization, this type of arrangement was evolved to better facilitate the movement of substances as the plant evolved from the water systems to a more land plants. So like those plants which are growing in land, they have to, and they are very big plants like angiosperms and gymnosperm, so they have to conduct water and nutrients to a great height. So this conjoint vascular system provides them a better system. It is in terms of anatomical structure also, it is considered that it has a more better organization. So definitely the root stem transition takes place, and there is a particular zone which is a very short transition zone which converts the root vascular system into a stem vascular system. Now 4 types have been observed, like A, which is also known as mirabilis type, then B is cucurbita type, then Latharis type, because these are typically found in these organism, although many other organisms are also showing this type of arrangement. Now what happens, let us try to discuss these transition. Now suppose in type A, mirabilis type, what happens? Suppose we start with the root system, okay, from root it has to convert into a stem. Root has a radial arrangement, okay, So what happens in the first type? Now this is the xylem and this is the phloem in discrete patches. Now what happens? This xylem radially bifurcate into two parts. Okay, they radially bifurcate into two parts. Okay. You can see that the xylem has divided into two, and this xylem, see here protoxylem is on the outer side, so they actually flip in the lateral side, at taking a 180-degree turn, so that this portion comes on this side and this portion comes on the other side. And what it is that they flip and they join the phloem. So how it is happening? Let's try to see. Okay. So they flip and they turn in the lateral direction, and these two, one part from each xylem, they will come and they will join this phloem like this. See, you can see this is the phloem and this is the xylem. So both the parts come together, they align together, forming a conjoined vascular bundle and the endarch situation. Because the xylem has also flipped to 180-degree, so their position has also inverted and they have now joined just below phloem. So now they have become conjoined. So in this way, these four types, you can see it is explained in all these figures. See, this I was talking about. This is the mirabilis type or type A, where you can see these xylems have bifurcated and they have flipped and they have joined together. Now in this case, the number of xylem patches or the phloem patches, the same number of vascular bundles are formed. So here also 4 vascular bundles and here also 4 patches. In the second one, which is also known as a cucurbita type, in this you can see 2 xylem patches are there and 2 phloem patches are there. Xylem patch will bifurcate, okay, and in this phloem will also bifurcate, means xylem both will bifurcate, and each patch will join the other patch. See, this patch of xylem is joining with this patch of phloem. Similarly, all the 4 patches of xylem will join with all the 4 patches of phloem and they will flip 180-degree. So definitely this will come to this side. So this will flip in this way, and finally 4 vascular bundles will be formed. So in this case, starting with 2 patches, but finally we are getting 4 vascular bundle. In the third type, you can see here, we are starting with 2 patch. Here, in this case, phloem is dividing, however xylem is not dividing. So in this case, you can see, but definitely xylem has to flip because exarch condition has to be converted into endarch. So it is flipping 180-degree, but it is not bifurcating. However, phloem is bifurcating, and in this, one xylem will divide with 2 of the phloem patches, and finally you will get a vascular bundle which will be almost with the same number of vascular bundles as was the patch in the root system. Okay. Now the last one, in this you can see here that 2 of the xylem are bifurcating. Okay, 2 of the xylem patch is as such. However, 2 of the xylem patches are bifurcating. See this bifurcating. Although all the 4 are flipping, these 2 bifurcated form as well as the one which is not bifurcated, they all are flipping. Okay, all are flipping to 180-degree turn. They are taking in the lateral direction, and then the 3 this xylem patch. Okay, 2 of the phloem patch will join together. Okay, that means out of 4, finally 2 phloem patch will be there and 3 xylem patches. Okay, one from the bifurcated one, 2 bifurcated one, and this one which is not bifurcated, they will go and they will form, they will join with the 1 phloem patch. Okay, that means 2 phloem patches are joining. Okay, they are making 1 phloem patch, and out of the 6 these xylem patches, means 2 which have been bifurcated, 3 are joining with each phloem, and finally you are getting 2 vascular bundles. So in this case the number of vascular bundles is reduced to half. So there are conditions where the number of vascular bundles are same. Okay, same number of vascular bundles, in which it has been doubled. Okay, the number of vascular bundle is double. Okay, from 2 it has become 4. In this case also it remains same. However, the pattern of division is slightly different. In this xylem was bifurcating, in this phloem is bifurcating. In this xylem, and in this case you have half the number of vascular bundle. So this is all about the transition. Now in the last I would just like you to revise whatever has been taught in the root section, and for this revision it's better you just try to understand what is there in a dicot root, what is the composition of cortex, then endodermis, the number of xylem and phloem bundles, then what is the nature of cambium, secondary growth and pith in dicot root as well as in monocot root. So in this way it will help you in better understanding of all the concepts that we have discussed. So this is about stem we have already discussed, root, and then in the next lecture we will try to cover up with the leaf anatomy. So thank you all for today.